Good afternoon, and thank you for the organizer for having invited me to speak tonight. I have to disclose that I am a PI in the DBV SMILEY trial, in which we try the EPIT in eosinophilic esophagitis, and my funding otherwise come from the American Academy AFED, an institutional grant. So eosinophilic esophagitis is an inflammation of the esophagus. It's defined by the presence of at least 15 eosinophils per high power field in the esophagus. The esophagus typically is devoid of any eosinophils. And it's isolated to the esophagus, doesn't have any eosinophils. You don't find them by definition in other part of the intestinal tract. Who are the patients who get eosinophilic esophagitis? They're typically very atopic patients. Up to 90% of the patients have other comorbidity with other atopic disease, mainly allergic rhinitis, but also food allergy. In some study is up to 20-25% of IgE-mediated food allergies. There is a genetic component in EOE. So the risk, if you have a siblings, is over 50 times to have the disease compared to 2 if you have asthma. But monogenetic studies shows that the genetic component increase the more the genetic makeup is similar. So it doubled from dizygotic twin to monozygotic twin. But even in monozygotic twin, the concordance is only 40%, and the twins who are dizygotes, they have much more concordance than siblings, even if the genetic makeup is similar, but probably the environmental exposure is much similar in twins than in siblings. And this environmental component is what makes only 40% of the monogenetic twins to have the disease. So which are the triggers of EOE? Even if environmental allergens and infection have been reported as cause of EOE, those are more anecdotal and we have less body of evidence. The majority of body of evidence that is most convincing is that food allergens are one of the major causes of EOE, both in adult and children. And this mainly comes from the clinical practice in which elemental diet, we know, treat the disease both clinically and pathologically in virtually in all patients. And then diet that exclude either all the major food allergen called 6-food elimination diet, or diet that they're more eliminating targeted based on the patients, still can make the disease goes away in about 70% of patients, and diet is an approved treatment of EOE and accepted in all the guidelines, both in adult and children. And one of the food that we see both in adult and children that is one of the major cause of EOE is milk. And this now it's approved also in adult. When we see the type of inflammation that is present at the site of the esophagus in patients with EOE, it's a typical atopic inflammation. There are the typical cytokine of atopy, interleukin 13, interleukin 5, interleukin 4, and the cells that they're present at the level of inflammation are the typical cell of the atopic inflammation. They are basophil, they are mast cells, they are T cell, Th2, they are basophils. And this is an old study from Stroman, older than almost 15 years old, but this has been proved by many different groups throughout the world. And these are data from Paul Aboni about mast cells that they're increasing patients with EOE, and the markers of mast cells are also increased. So how do you get from food allergen or the environmental exposure to this Th2 inflammation? Some clue came after studying the genetic of this patient. We see that the genetic component is important, so there has been done study both through using the candidate gene approach and genome-wide association to try to find target that explain why this inflammation happens. One of the earlier study from the Rothenberg group, they looked first at the RNA level using microRNA throughout the whole in the epithelial cell from patients with EOE, and they found an increased level of eotaxin 3, about 50 times higher than in control patient. And then when they went to look at the gene of eotaxin 3, they found a single nucleotide polymorphism that it was more frequent in a subgroup of patients with EOE compared to the normal patient. That prompted to do bigger study using more powerful tool to see if other targets could be found to speed up the research in the pathogenesis of this disease. And our group, led by Dr. Spergel, with the Cincinnati group and in collaboration with many other centers throughout the states, accumulated about 500 patients, and the Center of Applied Genomics at CHOP ran a genome-wide association study between patients with EOE and control, and found that there was a SNP that it was less expressed in patients with EOE. So there was a protective SNP that came up and was protecting from EOE, and that SNP was in the TSLP gene, and in accordance, patients with the protective SNP had less TSLP expression in their esophageal biopsies. So that led to focus the attention of TSLP, and TSLP is a cytokine secreted again by the epithelium, and it's known to induce a Th2 inflammation in the sites where it gets secreted, and it's known to be elevated in other atopic diseases such as asthma and atopic dermatitis. And what TSLP does, it does induce Th2 cytokine by acting directly to the major player of the Th2 inflammation. But one of the major roles, like Cecilia have shown, is to tell the dendritic cell to induce a Th2 inflammation once they present the antigen to the T cell. So they skew the T cell response towards the Th2 response and not towards the Treg response, which is the natural response. So the other question was if this TSLP had any relevance biological in EOE pathogenesis. So the Artis group, when it was at the University of Pennsylvania, developed a model of EOE in which mice sensitized through ovalbumin after being pre-sensitized with an analog of vitamin D were developing a disease similar to EOE, which included also the food impaction. And this disease could be stopped if the animal were treated with anti-TSLP, suggesting that TSLP was playing a major role in the development of the disease. But interestingly, also it showed that by using antibodies against the basophils, also the disease was stopped, suggesting that two players of the innate immune system, TSLP and basophil, were playing a major role in the development of the disease. And in human, this is our patients, showed that the patient with active EOE had higher level of eosinophils by definition, but also higher level of TSLP, and that the level of eosinophils in the biopsy correlated with the level of basophils, suggesting that this is not a phenomenon only present in mice, but also in human. And then his mouse study showed also that IgE were not essential for the development of the disease because mice that didn't have the immunoglobulin were still able to develop the disease equally to the other type of mice. And clinically, we know that this is not an IgE-mediated disease. There are lots of clinical data that kind of led us to the same conclusion. So skin prick testing and measurement of serum IgE in the blood of patients specific to food, and then designing a diet based solely on those data are not effective in designing an efficacious diet for these patients. Anti-IgE treatment with omalizumab also have been proven not to be effective in patients with EOE. And one of the major side effects of the oral allergy desensitization cause is the development in between 2% and 20% of the patient, depending on the study. Once the food is reintroduced because it's not causing any more IgE-mediated reaction, it actually patients then they ended up developing EOE. And we saw also the same phenomenon in about 30 patients that had previously IgE-mediated reaction to food, and they were able to reintroduce the food because they have outgrown the IgE-mediated reaction, which is a phenomenon quite frequent for milk and egg in children. But once the food was reintroduced, they ended up developing EOE to the same food. This is, for example, an example of a child that was in infancy allergic to milk in an IgE-mediated fashion, had an anaphylactic-like reaction. With time, the skin prick test went down. He was able to reintroduce the milk, which is the normal, natural history of IgE-mediated allergy to milk. But about 2 years after the reintroduction of the milk, he ended up developing EOE. So then our laboratory started to think of other ways that the cell could recognize the milk and start a recognition of the milk, and we focused our attention on a small population of T cell called invariant NK cell that they are able to that they have a special TCR receptor that is invariant and recognize a limited number of antigen presented through the CD1D molecule. And those antigen are not protein like happens in the regular IgE-mediated allergy, but they are lipids. And once activated, the iNKT cell, even if they recognize a specific antigen, they behave a lot like innate immune cell. They release a lot of Th1 and Th2 cytokine without requiring a clonal expansion. So the lipid that they are able to activate the iNKT are usually sphingolipid or glycerol-based lipid. And in our experiment, we tried to see if a sphingolipid, the milk sphingomyelin, was able to activate this cell. And we saw that in normal donors was able to activate the cell and make them proliferate. Then we wanted to see that if this cell had any role in EOE, and we measured their level in active EOE patient in their biopsy and in the peripheral blood, and we saw that those levels were reduced in the peripheral blood and were increased in the biopsy, suggesting that during an inflammation, those iNKT could migrate in the biopsy, and this migration wouldn't happen in patients with controlled disease or with or patient without the disease. And this same phenomenon has been described in adult patients where with active disease against the iNKT were high in the biopsy and low in the periphery. And we saw that the iNKT in the periphery had lower level of chemokine receptor 3 and 5 and 4, which are the receptor for eosinophilic chemotactic factors such as rantis and eotaxin 3, suggesting that these could have been depleted, this peripheral population could have been depleted of those iNKT that have been called at the site of an eosinophilic inflammation. We also saw that patients with active EOE, once we stimulated the iNKT with milk lipid, they produced more Th2 cytokine than the patient who had the controlled disease or those patients that didn't have EOE. suggesting that this cell could be also contributing to the Th2 inflammation at the site of the eosinophilic esophagitis. But when we have all these Th2 cytokines secreted by different cell type, what happens next that can favor a chronicization and an expansion of the inflammation? So Th2 cytokine have been shown in atopic dermatitis to be able to reduce the barrier function and makes the skin much more permeable. And in a follow-up study to the genome-wide association study that we have done before, where TSLP SNP was identified, by studying about 800 patients, we found that another SNP in another gene, the gene of calpain 14. And this was a study that came out right after another study that showed exactly the same result, but it was led by the Cincinnati group. And those were two independent studies using the independent patients and showed the same hit on calpain 14. And this hit was interesting because these are the data from the Cincinnati group. They demonstrated that the esophagus is the main producer of calpain 14. So this is a molecule specifically expressed in the esophagus, and interestingly is modulated by Th2 cytokine. So the interleukin 13 increases the calpain 14, and patients with active disease, which are known to produce Th2 cytokine, had a higher level. The exact role of calpain 14 in the esophagus is still not completely known. However, calpain 14 is a protease, and as the proteases, when it's elevated, cause damage to the epithelium. And we know, and there are other molecules that have been shown to be reduced, that they are protecting the epithelium and have been shown to be reduced in EOE, such as desmoglein 1, that in patients with EOE have been shown to be reduced, and interleukin 13 again reduce the production of desmoglein, and once those molecule protective are not there, they cause an increased permeability. And today at the abstract session, there were data that SPINK5, SPINK7, and they are also all antiproteases that they help to protect the epithelium. They are lacking in EOE, and they favor the permeability of the epithelium. And once this permeability is impaired, like in atopic dermatitis, that favor has a twofold consequences. One, favor the allergen to come into contact with the immune system, and two, to continue the epithelium to secrete pro-inflammatory cytokine that create a vicious circle and a sensitization. So in what we think it's possible that can happen in EOE is that we have a genetically predisposed individuals which are predisposed to produce more of this Th2 pro-inflammatory cytokine and chemokine from the epithelium. Once those get released by the epithelium, they call all the major player of the Th2 inflammation, and now those players are there secreting Th2. These instruct the dendritic cell to induce once they see the allergen, the protein allergen, they instruct the cell, the Th2 cytokine and TSLP, to stop the regular Treg response and instead do a Th2 response. And as Cecilia has shown, once this starts to happen, is a self-fulfilling prophecy, because more Th2 cause more sensitization and more Th2, and then they inhibit Treg. So in conclusion, and this Th2, then they favor the epithelial dysfunction, and hence more allergen comes in. So we think that one model in which the innate immune system may play a big role would be the epithelium and cell of the innate immune response, such basophil muscle and iNKT that produce Th2 cytokine, then they favor this local sensitization to the protein allergen via the T cell, and we're working now on demonstrating that T cell are really the major player in recognize the antigen and creating the inflammation.